Welcome to Bio 112 Online, your home for biology education. Today's topic, the circulatory system. In the last lecture, we covered the cardiac cycle, going from diastole to atrial systole to ventricular systole, and then back to diastole. And the whole purpose of this was to pump blood throughout the body. So let's take a quick moment and think about that blood for a moment and what it's made up of. The blood is going to be made up of red blood cells or erythrocytes. These are going to contain hemoglobin and are going to be responsible for carrying gases in the blood. Other interesting aspects of these is that they do not have a nucleus when fully mature. This is in contrast to the white blood cells, such as the leukocytes. We covered these in our discussion of the immune system, so we won't dwell on them. The next main component is going to be the plasma. The blood plasma is going to be the solution, i.e. the water, and all of the solutes dissolved in it. This includes things like ions and also proteins. Many of these proteins will have a diverse array of functions. Think back to our discussions of the complement proteins and antibodies in the immune system. It'll also contain a whole variety of other compounds, such as glucose, such as hormones, many of the other things we've been talking about. Finally, we want to talk about the platelets. The platelets are going to be responsible for helping to form clots within the blood to prevent loss of blood from the body. The next thing that we want to do, now that we have an idea of what blood is to trace that path of blood as it flows through the heart. Within that flow path, we have two main circuits we want to concern ourselves with. The first of these is going to be the pulmonary circuit. In the case of mammals, we have two pulmonary circuits, one on the left, one on the right. These are going to be the circuits that bring the blood from the heart to the lungs and then back again. The capillaries within this circuit are going to be responsible for gas exchange with the environment. The other main circuit we need to concern ourselves with is going to be the systemic circuit. We have the anterior systemic circuit, which will feed blood to the upper extremities of the body, where the posterior systemic circuit will feed blood to the digestive system and then the lower extremities of the body. It's useful in this case to go through and track the movement of blood through the circulatory system. We're going to start at the right ventricle. As the right ventricle contracts, it pumps blood through the pulmonary arteries to the lungs. Keep in mind in this case that the blood flowing to the lungs at this point still does not have much oxygen. It then moves to the capillary beds of the lungs where it picks up oxygen and unloads carbon dioxide. That now oxygen-rich blood returns to the heart through the pulmonary veins and enters the left atrium. That oxygen-rich blood then enters the left ventricle and is pumped out to the systemic circuits through the aorta. There's then a branch to the anterior circuits through the coronary artery, or to the aorta going to the posterior circuits. In either case, the blood will unload that oxygen and load up with carbon dioxide. That oxygen-poor blood coming from those capillaries will then move towards, in the case of the anterior circuit, to the superior vena cava, or in the posterior circuit to the inferior vena cava. In both of those cases, that blood will move back to the heart, enter through the right atrium, and then back to the right ventricle where we started. What I'm going to suggest for you guys is to go through, take a look at this circuit, and be able to replicate it. As it's traveling through these blood vessels, we should think about what are the commonalities and differences between arteries and veins. Quite often, you would think about arteries and veins in terms of whether or not they're carrying oxygen or non-oxygenated blood. But we just sort of dispelled that myth right now because we said in the case of the pulmonary arteries, it's carrying oxygen-poor blood. and the pulmonary vein is carrying oxygen-rich blood, and then that reverses in the systemic loop. So what really is the difference between an artery and a vein? Arteries and veins are composed of concentric layers of connective, smooth muscle, and epithelial tissue. The smooth muscle within those arteries and veins will respond to numerous nervous signals and hormonal signals, which will allow them to contract or relax. This plays a role in the control of blood pressure and can also control blood flow to different parts of the body. The innermost layer of those blood vessels is referred to as the endothelium. The endothelium is going to be a single layer of flattened epithelial cells. It's going to be responsible for sealing off that blood flow and keeping the fluid from leaking out into the rest of the body. Those are the main commonalities between arteries and veins. Those are the main commonalities between arteries and veins. So what are the differences? Arteries and veins will have a few major differences. The first of which, in the case of the arteries, is that they're about 3 times as thick as the veins. This is because the blood flowing in the arteries is under much higher pressure, and the blood vessel requires the additional layering of the artery walls to contain that pressure. Veins are going to be of much lower pressure, to the point at which they have valves located within them, and those valves help to move that blood along and prevent it from flowing backwards and keep it flowing in one direction towards the heart. In betweenst those arteries and those veins are going to be a number of smaller blood vessels. These include the arterioles, which are very small versions of the arteries, the venules, which are going to be very small versions of the veins, and finally the capillaries. The capillaries will be structurally different though. Capillaries are going to be thin-walled blood vessels that supply resources directly to the tissues. As a result, they do not have any muscle tissue, and they don't have much in the way of those connective tissue walls. They're simply there as a way to then be thin and deliver the resources to the parts of the body that need it. The main thing that the capillaries are responsible is exchange. As a result, one of the main things we discuss when we're talking about exchange with the environment is all about surface area. If you were to take a look at the sum cross-sectional area of the capillaries compared to that of the venules, the arterioles, the arteries, and the veins, you'd find that the sum of all the capillaries leads to a larger total cross-section in those capillaries, which maximizes the amount of exchange. As blood flows into those capillaries, because there's so much cross-sectional area, then the blood has a tendency to slow down and move much more slowly during the transit through the capillaries relative through the arteries and the veins. The blood pressure also has a big change as you move from arteries to arterioles to capillaries to venules to veins. In this case, the blood pressure will have a tendency to decrease along the length of that circuit. The main reason for that decrease in pressure is going to be a certain amount of resistance to the blood flow. There's going to be a certain level of friction that goes on between the fluid flowing through the blood vessel and the walls of the blood vessel, i.e. the surfaces of those epithelial endodermal tissues. As you have that friction going on, that will be less evident as you're moving through wider vessels. But as you move progressively smaller and smaller vessels, the cross-sectional area decreases but then the surface to volume ratio will then increase, leading to greater and greater amounts of friction. So therefore, the diameter of the blood vessel, the length of the blood vessel, and the blood's viscosity will all play a role in generating that friction and slowing the blood down. It will dissipate energy in that case and decrease the overall pressure. Now, when we talk about blood pressure, we should be a bit more specific. The blood pressure in general within the body is going to be controlled by several main factors. The first of these is going to simply be the volume of blood. If there's more fluid packed into your circulatory system, it will have to be at a higher pressure, unless your blood circulatory system actually expands out. The next main factor is going to be the cardiac output of the heart. How fast is the heart beating? What is the rate of the heartbeat? And in addition, the stroke volume. With each beat, how much actual fluid is being pushed through. Therefore, the cardiac output is a total volume of blood per unit time. The other main factors we kind of hinted at is what is the ability of those blood vessels to expand. The elastic force applied by the blood vessel walls will have an effect on the blood pressure. Finally, a change in actual size of those blood vessels, either through the process of vasoconstriction or vasodilation, can also play a critical role in determining your blood pressure at any given time. Vasoconstriction, as you'll remember from the beginning of this course, is the contraction of smooth muscle in the blood vessels to reduce the cross-sectional area of an artery or vein. This is going to be useful on a local level, directing blood into one area or another. Typically, in that case, vasoconstriction prevents blood from flowing into an area by reducing the cross-sectional area of that blood vessel. Vasodilation does the opposite, relaxation of the smooth muscle in the blood vessels to increase the cross-sectional area and therefore allow more blood to flow through. At a more systemic whole body level, if your entire body starts increasing its vasoconstriction, you can inherently raise the blood pressure of the entire body. In addition, if your body is exercising heavily and you have a high heart rate and high cardiac output, then your blood vessels might go into the process of vasodilation, which expands out the blood vessels to accommodate the increased flow and allow you to deliver the blood to the parts of the body that need it. So we're all familiar with going to the doctor's office and hearing about our blood pressure, and they usually report it as two numbers. It'll be something like 120/70. So what do these numbers really mean? The two numbers are going to represent the systolic and diastolic pressure within the blood. You recall those terms, diastolic and systolic, in terms of the heartbeat. We said systole was going to be when the heart is contracting, particularly in this case, when the ventricles are contracting. The systolic blood pressure in the arteries is going to be the pressure during the contraction of the ventricles. It's going to be the highest point of blood pressure during the cardiac cycle. This occurs because the blood being pressed into the arteries by the ventricles is going to be moving into those arteries faster than it can leave through the arterioles. Therefore, there's this backup of blood causing the arteries to stretch a bit. After that pulse of blood passes through, those arteries start to shrink down and shrink down and shrink down until the next pulse comes down and starts expanding them out again. But they're never quite fully deflated and returned back down to a flaccid state. This lowest pressure, this pressure before the next pulse hits, is referred to as the diastolic pressure. It is being generated now by the elasticity of the walls of those arteries, still in the process of contracting back down before the next blood pulse hits. So we can feel this whenever we're talking about blood pressure. You can actually go to your own wrist and you can start to feel your pulse. That little tick you feel with your pulse, that little bump, 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 is really the systolic pressure moving through. So how do we go through and measure this systolic versus diastolic pressure? Well, whenever you go to the doctor's office, they'll go through and they'll wrap a little collar around your arm. The device that they wrap around your arm is referred to as a sphygmomanometer. Man, try saying that 5 times fast. You just tried it, didn't you? Okay, try it again. Okay, moving on. So the sphygmomanometer is an inflated cuff around your arm, and it inflates to a pressure enough to push down at a higher pressure to cut off your blood flow in that artery. Once it completely occludes that artery, no longer can you have blood flow. It will then start easing up on that pressure. So once you hit the systolic pressure, what happens? You have just enough of a push of blood that it can lift up, let some blood through, and then slam back down shut. That can be heard in a stethoscope placed against the arm or felt by a device as a little thump. So what that device is doing is, as soon as you get to that point where the blood pressure equals the pressure of that cuff and causes that little thump, thump, thump to start, it'll start counting that. And that becomes the systolic pressure. It'll keep on measuring. You have a pulse of blood that moves through, lifts it up, and then thump. Lifts it up and thump. Until the pressure of the cuff is no longer sufficient to even occlude the blood vessel during diastole. The thumping will stop. And that becomes the diastolic pressure. And it's the point at which the blood again flows freely. It should be noted here that blood pressure is being measured in the arteries. Blood in the veins is at a much lower pressure, as we said, and may have difficulty returning to the heart by pressure alone. One of the key ways in which your body helps return that blood to the heart is going to be through the use of those valves. We said that those valves are similar to the valves found in the heart and prevent backflow. This becomes useful whenever we're moving our skeletal muscles around. As we're moving our skeletal muscles around, our skeletal muscles are squeezing our veins. As you squeeze the blood in those veins, you can get it to push together. Blood will want to move in both directions, but those valves will prevent it from moving backwards, meaning that you only have a one way to go, a linear flow of blood back to the heart. In addition, contraction of smooth muscles in the walls of the veins and changes in the thoracic cavity pressure while you're breathing can also assist to move blood from the extremities back to your heart. Let's take one more look at these capillaries, these small, thin-walled vessels that supply resources directly to the tissues, and look at the ability of these capillaries to exchange. Keep in mind that capillaries aren't all active throughout your body all the time. At any given time, only about 10 to 15% of capillaries will have blood flowing through them. This blood can be allowed into the capillaries via the precapillary sphincters. The precapillary sphincters are going to be small muscles that, when they contract, prevent blood from flowing into the capillary bed. So 4 times when your body and that particular tissue is actively metabolizing, those capillaries were open, allowing the blood to flow through. However, if those tissues are going a bit dormant, aren't being used as much right now, those capillary sphincters can tighten, moving blood to areas that actually need it. When those capillary beds are available and are open, then the method of exchange happens on one of several levels. The first of which, small molecules such as oxygen and carbon dioxide, especially if they're nonpolar, can simply diffuse through the cells that line the capillary. In addition, for larger molecules, such as proteins, vesicles can be formed in those epithelial tissues to pull those vesicles of proteins and transport those to the other side of the cell and place them into the interstitial fluid. The interstitial fluid, in this case, is going to be the fluid that surrounds all of the cells. You can have a third type of flow due to leaks within the tight junctions in those epithelial endodermal cells, which will allow for small molecules such as sugars, urea, and ions to move from the blood into the interstitial fluids. In addition, you have what's referred to as a bulk flow of fluids between the capillaries and the interstitial fluid. This bulk flow of fluid will be driven by differences in blood pressure from the arterial end of the capillary bed to the venular end of the capillary bed. At the arterial end of the capillary bed, the sum of the solute and pressure potentials within the capillaries exceeds that of the sum of the solute and pressure potentials in the interstitial fluid. As a result, because of the water potential, water will then move into the interstitial fluids, delivering resources to the cells that need it. Once you get to the venular end of the capillaries, you get to a point where the pressure has dropped enough that the sum of the solute and pressure potentials of the capillaries is now lower than the sum of the solute and pressure potentials in the interstitial fluid. As a result, fluids will have a tendency to flow from the interstitial fluid back into the capillaries, with some additional loss of fluid entering the lymphatic system. The movement of blood in the capillaries is critical for the delivery of resources to the body and is also essential for exchange of gases with the lungs. But that is the topic of the next lecture. So until then, stay safe, stay healthy, and I'll see you later. That collar and the associated measurement device is referred to as a sphygmomanometer. Man, I just butchered that. Is referred to as a sphygmomanometer. The sphygmomanometer, sphygmomanometer, yeah! See, this is going to be what goes in the bloopers reel. So until then, stay safe, stay healthy, and I'll see you later.